I am Mark Nagel from Trudell Medical International, and this presentation covers the use of a sectional adult nasal airway model for the evaluation of nasal delivery devices and administration techniques. The nasal cavity is an attractive portal of entry for aerosol and spray medications, either for topical delivery to the nasal passageways or for access to the brain via the olfactory bulb bypassing the blood-brain barrier. However, its complex internal geometry varies widely in detail from one individual to another, especially in the sinus cavities. Nasal models with anatomically accurate internal structures have been developed in recent years to provide realistic information regarding likely deposition of active pharmaceutical ingredients in the process of developing improved delivery systems. Guimet and Gagliano were one of the first investigators to develop anatomically accurate nasal replicas that could be used to evaluate the likely deposition of nasally administered sprays and aerosol products. Their approach avoided the use of cadaver-derived models where tissue collapse post-mortem had been identified as limiting their effectiveness. In this study, two variants of a sectional adult nasal airway model were developed based on data from an adult MRI scan. The study purpose was to visualize deposition patterns and measure regional deposition from a nasal nebulizer. Both models have the exact same internal geometry. However, Model 1 was longitudinally segmented and transparent to enable visual observation of a color-changing product for qualitative assessment of deposition from the nasal nebulizer. The second model was made from a sintered nylon to allow for quantitative assay of a test active pharmaceutical ingredient, in this case budesonide, without the risk of leachable contaminants. The second model was segmented horizontally into an anterior, middle, and posterior passageway. SAR gel was applied to the interior surfaces of the model before a spray containing budesonide was delivered to the nasal vestibule by means of the nasal nebulizer. This compound becomes purple when it comes in contact with liquid water, which in this case accompanies the suspension of budesonide particles in the nasal spray. Most of the color development occurred in the middle section of the model, providing qualitative evidence that the budesonide droplets also deposited in this location. Laser diffractometry provided a non-invasive way to quantify the water droplet size distribution emitted by the nasal nebulizer. This distribution is more important than the underlying size distribution of the budesonide particles in the nebulized suspension, as the water droplets convey the budesonide particles to where they deposit in the nasal cavity. In this particular instance, droplet size distributions were made by the SprayTech laser diffractometer at a 6 centimeter working distance. This time-resolved chart shows the evolution of the aerosol from the nasal spray. Inevitably, the 90th percentile data, shown here as magenta, contains more noise than the corresponding 50th percentile shown in green, which in turn is shown noisier than the 10th percentile shown in blue. However, the volume mean diameter corresponding to the 50th percentile has been chosen as being most representative of the measure of where the bulk of the accompanying budesonide would deposit in the model. Volume mean diameter values in the 50 to 60 micron diameter range are expected to penetrate the nasal vestibule and deposit in the middle region of the model. From reported modeling studies of droplet transport through the nasal cavity, such droplets are unlikely to penetrate further to reach the nasopharynx, represented by the posterior segment of the model, where droplets that deposit there are typically closer to 10 microns in aerodynamic diameter. The budesonide particles in aqueous suspension would be expected to be transported within the water droplets following spray generation at the nozzle exit of the nasoneb nebulizer. Their final deposition location should match closely with that for the water droplets as observed with Model 1. Deposition of budesonide in the middle segment of Model 2 would therefore be the anticipated outcome. The measurements reported in this presentation provide a preliminary validation of these identical anatomically correct models of an adult nasal cavity. The transparent Model 1 allows for rapid visual interpretation of the likely deposition profile. The opaque Model 2 is suitable for quantitative measurements of drug particle deposition by an appropriate assay technique. Further studies will include evaluations with nasal sprays and aerosol sources, producing a range of different size distribution profiles.